We will learn the chapter 3, so epigenetics in Schizosaccharomyces pombe. Schizosaccharomyces pombe, we call it as fission yeast. So today, first we will learn the fission yeast life cycle, and also we will learn the heterochromatin region in the centromere, and also we will learn the heterochromatin formation mechanism. So finally, we will learn the facultative heterochromatin. Okay. So in this slide, I will explain the first subject, life cycle of fission yeast. So fission yeast, we call it as, in Korean, we call it as the 분열효모. So fission yeast is a eukaryote. And fission yeast, they contains 3 chromosomes, and for example, this is chromosome 1, chromosome 2, and chromosome 3. So each chromosome have the heterochromatin region, especially the telomere region and the centromere region. Especially the telomere and centromere region is the heterochromatin region. And also especially is the chromosome 2 has the mating type gene. It's mating type locus. Okay? Chromosome 2 has mating type locus. So usually the chromosome 1, 2, 3 is existed inside of the nucleus. Okay, so there the genome size is about 14.1 megabase pairs, and also they have only 5,000 genes. And their split time is that 2 hours at 30 centigrade. So one fission yeast become the 2 fission yeast after 2 hours later. Okay, and also their life cycle is composed of the haploid and diploid. So they are a mixture of haploid and diploid. So we'll see some detailed information in next slide and also next slide. And also they have the regional centromere. So regional centromere is the most popular form of centromere. So from the fission yeast, so for example, budding yeast, they are very low classes in the eukaryote, so they don't have the regional centromere. But from fission yeast, they have the regional centromere. And also especially the fission yeast, they have the histone H3K9 methylation. For example, so histone H3 is composed of several amino acids, especially the 9th location. K means that the simplest code of the lysine. So lysine, so in H3 histone 9th location, there is a lysine is there. So lysine is usually the methylated in fission yeast. Okay, so we will see some detailed information in the later. Okay, and especially the SUI6 protein, we call it as a HP1 protein. So heterochromatin protein 1. So this SUI6 or HP1 protein is bind to methylated histone H3K9. After that, just this protein is decodes the information of methylated histone H3K9. Okay. So in this slide, I will explain some detailed information of haploid status, and next slide I will explain the detailed information of diploid status. Okay, so haploid is we call it as vegetative cells. In Korean, we call it as 영양성장적 세포. So this haploid is composed of the plus mating type haploid and minus mating type haploid. How about in budding yeast? We already learned that budding yeast is composed of the A haploid or alpha type haploid. Okay, a little bit different. And also the mating type region is also present in the heterochromatin region, and also this mating type region is located within the chromosome 2. Okay. Now I will explain the cell cycle of haploids. So this is the cell cycle of the vegetative cells. So they are composed of the interphase, and they are also composed of the M phase. Okay, the interphase is composed of the G1 phase and S phase and G2 phase. So G1 phase is composed of G1 and S and G2 phase. So usually in the cell cycle, the interphase is composed of G1 and S and G2 phase. So usually the time of the G1 and the time length of the G1 and G2 is nearly similar to each other. But in the fission yeast, G1 is composed of the 10% of whole cell cycle, but G2 is composed of 70% of whole cell cycle. Okay. So during S phase, so DNA is replicated. Okay. And also M phase is composed of the prophase, metaphase, anaphase, and telophase. After telophase, so there is the cytokinases. It means that one fission yeast becomes the two fission yeast. Okay. Okay. So this is the cell cycle of haploid, and next we will cover the meiotic cell next slide. Okay. In this slide, I will explain the diploid status. So diploid means that meiotic cells. So meiotic cell is initiated by the conjugation. Conjugation means that when fission yeast is very limited in nutrition, at that time two nearby fission yeast are physically combined with each other to form the diploid. So after that they physically combine to each other, and after that they form the diploid status. Okay. After that there is also cell cycle of meiotic cells. It's composed of the meiosis 1, and after that meiosis 2. After that there is a sporulation. So sporulation means that 4 haploid spore can be made from sporulation. Okay, so diploid cell we call it as a meiotic cell. So meiotic cell is initiated by the conjugation. So after the conjugation, their status becomes the diploid. After that, this diploid cell doing the meiosis procedure. For example, meiosis is composed of the meiosis 1 and meiosis 2. After that, the sporulation is happen. So each cell become the one haploid spore. Okay, so finally there is a 4 haploid spore becomes. Okay. So in this slide, I will explain the second subject, the heterochromatin in centromere. Okay, let's start from the heterochromatin region in fission yeast. So we can easily see in the centromere region, we can easily see the heterochromatin region, and also telomere region. We also can see the heterochromatin region, and also in chromosome 2, there is a MAT locus is located here, and this locus is also within heterochromatin region. And the life cycle of the fission yeast is composed of the diploid status and also haploid status. And their life cycle, most of their life cycle is the haploid status. So that's why this is due to because the gene locus involved in meiosis is located in the within heterochromatin region. So the meiosis starting from the conjugation and meiosis 1 and meiosis 2 sporulation is not frequently happen in Pigeon yeast because the gene locus involved in meiosis is located in the heterochromatin region. Okay, and also, so we will see some detailed information of the heterochromatin in centromere. So heterochromatin in centromere is observed in Pigeon yeast, observed from Pigeon yeast. Okay, previously we learned that budding yeast, they have the point centromere, but point centromere is about 125 base pair, but they don't have any heterochromatin region. However, so budding yeast, they show that regional centromere. So regional centromere is most popular form of centromeres. But you see here in the center there is the CEMPA binding region, and next to the CEMPA binding region, so there is a heterochromatin region. Heterochromatin region is existed. Okay, so this is the Pigeon yeast centromere. Okay, and also molecular mechanism is similar to that seen in higher organism. For example, this is the Pigeon yeast. At the center there is the CEMPA binding region, and the neighboring region there is the heterochromatin region. And fruit flies also show the same pattern. For example, at the center there is the CEMP binding region, and the next to the center there is heterochromatin region and heterochromatin region. And also human also show the same pattern. This is the human's centromere. So, and at the center there is the CEMPA binding region. So next to the CEMPA binding region there is the heterochromatin region. Okay. So in this slide I will explain the centromere structure. Okay, centromere is composed of the central region, central domain or central region, and also they are composed of the out region. Okay, let's start from the central region or central domain. So central domain is composed of the central core, we call it as the CNT, and also composed of the innermost repeats, IMR, innermost repeat, left innermost repeat, and right innermost repeat. Again, okay. And also, actually, in the central core region, CNT region, we call it as the CNT region, and they are also composed of the left IMR region and the right IMR region here. So actually, in the central region, central core region, this region is the kinetochore binding region. So this is the kinetochore. Okay, this is kinetochore binding region, and also this is the CNT region is also CEMPA binding region. So what is CEMPA? So usually the normal chromosome region, so they are composed of nucleosome. So their nucleosome is composed of the 8 histone, which is composed of the 2H3, 2H4, 2H2B, 2H2A. But especially in the centromere region, there is some variant existed. The nucleosome, we call it as CEMPA nucleosome. So instead of the H3 histone, there is a CEMPA histone variant is located here. So this nucleosome, we call it as the CEMPA nucleosome, and normal nucleosome, we call it as the H3 nucleosome. Okay, so this CNT central core region is also CEMPA nucleosome binding region. Okay, And also this CNT region, normal H3 nucleosome also can bind to the here. And so usually the microtubule can bind to the CEMPA nucleosome or the H3 nucleosome, which is located in the kinetochore. Okay, and also out region is we call it as the heterochromatin region. Out region is composed, this is the out region and this is also out region. So out region is composed of the out repeat. So we call it as out repeat. So this is the left out repeat, OTRL, and this is the right out repeat, OTRR. So each repeat is composed of the DHDG sequence, DHDG, DHDG, and DHDG. So we will see some detailed information of DHDG in the next slide, okay? So in this slide, I will explain the experiment to reveal the function of the OTR sequence. In previous slide, we learned that in out region is composed of the out repeat and out repeat, okay? But in this out repeat is composed of DH and DG sequence, DH, DG, DG, DH, DG, DH sequence. So actually we don't know the exactly function of this sequence. So, okay. So in this experiment, so we use the part of the OTR sequence, especially the fragment of DG. So we use this is the full fragment of DG, but we use the part of the DG, we use the fragment of DG. So this fragment we call it as L5 element. So we insert a part of the OTR sequence, especially the fragment of DG. So we call it as L5 element to the euchromatin region. So this is the euchromatin region. So we inserted this sequence into the euchromatin region. So after that, we found that heterochromatin formation is initiated in this region. So after that, this region becomes the heterochromatin region, and also it induced gene silencing in this region. So based on this experiment, the conclusion was that maybe some transcription factor that bind to the unique sequence in the L5 element may play an important role to form the heterochromatin. So this is our tentative conclusion. Okay, maybe some transcription factor that bind to the unique sequence in L5 element may play an important role to form a heterochromatin. So this is our tentative conclusion. So in this slide, I will explain the experiment to reveal that transcription in the OTR region is important for heterochromatin formation. So this experiment scheme is nearly similar to the URA3 gene in birding yeast. Okay, so in the fusion yeast, we knock out the URA4 gene in the fusion yeast. So after that, we insert the URA4 reporter system. This reporter system can express URA4 gene. So insert URA4 reporter system in the OTR region of centromere. So this is the OTR region of centromere. Okay. So it means that these pigeon yeast can grow in a medium without uracil. So the function of URA4 is changes this one into the uridine monophosphate. So after that, this this uridine monophosphate will become the uracil. So even though we knock out the URA4 gene in the fusion yeast, because we inserted the URA4 reporter system, this reporter system can express the URA4 gene. So it means that this fusion yeast can grow in a medium without uracil. Okay, so after that we can generate mutant in which certain gene is deleted. So we used this fusion yeast. After that, just generate additional mutant in which certain gene is deleted. So if the deleted gene is not important for heterochromatin formation, so it means that heterochromatin formation will be happen in this near this region. So it means that there will be the no URA4 expression. So no URA4 expression means that no uracil synthesis. It means that there will be no growth in a medium without uracil. So no growth in a medium without uracil. So we also generate mutant in which certain gene is deleted. If the deleted gene is important for heterochromatin formation, it means that there will be no heterochromatin formation in the OTR region. It means that there will be URA4 gene can be actively expressed, so it induced uracil synthesis. So it means that this mutant clone, they grow in the medium without uracil. Okay, so through this experiment, people found that mutant in which RPB2 gene is deleted, we call it as a RPB2 mutant, and also mutant in which RPB7 is deleted, we call it as a RPB7 mutant. These two mutants can grow in a medium without uracil. So this two mutant can grow in a medium without uracil. It means that this two deleted gene is important for heterochromatin formation. So what is the RPB2 and RPB7? So actually this is RNA polymerase 2. So this is RPB2 and RPB7 is the other subunit of RNA polymerase 2. So it means that RPB2 mutant and RPB7 mutant so cannot initiate transcription in the OTR region. Okay, so maybe this mutant cannot initiate transcription in the OTR region. So it means that no transcription in the OTR region prevents heterochromatin formation in the OTR region. So maybe the conclusion will be the transcription in the OTR region is important for heterochromatin formation in the OTR region. So through this experiment, we revealed that transcription in the OTR region is important for heterochromatin formation, especially in the OTR region. Okay. So in this slide, I'll explain the fourth subject. Title is Heterochromatin Formation by RNAi. So we will learn the RNAi dependent heterochromatin formations. So when we learned chapter 1, we learned about position effect variegation. So at that time we learned flexibility of heterochromatin formation during S phase. So during S phase, DNA synthesis is happened, and simultaneously the mRNA synthesis happen. So DNA synthesis is released heterochromatin, except that it will make the euchromatin. And also mRNA synthesis is simultaneously happen. So at that time we briefly learned about the procedure like this. But now today we will learn some details, information of this RNAi dependent heterochromatin formation mechanism. Okay. Previously we learned that mRNA synthesis happens, it recruits RNA polymerase and also dicer. So dicer is generated siRNA, and siRNA R1 they bind to each other. So this complex is to move to where mRNA synthesis occurs. After that it recruits H3K9 methyltransferase. So H3K9 methyltransferase is made the methylation in the chromosome. After that it also recruits SUI6. So what is SUI6? We learned that in this chapter. SUI6 is bind to the methylated histone, and it decodes the information of methylated histone H3K9. So now we will learn some of the detailed information of the RNA dependent heterochromatin formation mechanism. So in this slide I will explain some detailed information of RNAi dependent heterochromatin formation. So this is the OTR region of centromere. It's composed of DH region and DG region. So in this region, bidirectional transcription is happen in OTR region of centromere. So it will generate non-coding RNAs. So non-coding RNA is they are composed of sense and antisense centromere RNA. So we call it as a sense non-coding RNA and antisense non-coding RNA. CEN means the centromere RNA. Okay. So usually the exosome. So exosome is usually bind to the most of non-coding RNAs. So it will digest it, all of them. But treatment, this protein specially bind to the remained non-coding RNA. So this treatment is to protect the non-coding RNAs from exosome. So this treatment bind to the remained non-coding RNA and processes RNA. So it will make the pre-RNA. So primer, so the full name is primer RNA, pre-RNA. It will generate pre-RNA. So usually this pre-RNA is there. So usually the ARG1 protein is bind to sense or antisense pre-RNAs. So it will make the ARG1 pre-RNA complex. So the ARG1 pre-RNA complex bind to the RNA template and use the pre-RNA as the primer. So use the pre-RNA as primer. Okay, except that it will recruit RDRC. So RDRC is composed of the RDP1 and CID12 and the HRR1. The main function of the RDP1 is RNA dependent RNA polymerase. So after it recruits RDRC complex, so after that actually the RPD1, so RNA dependent RNA polymerase is synthesize double-stranded RNA. So it will synthesize double-stranded RNA. After that, dicer DICE1 protein. This protein is cleaves the double-stranded RNA to the siRNA, double-stranded small interference RNA. So it was very long, but now this dicer cleaves dsRNA to siRNAs. Okay, so at that, so ARC complex is composed of the ARG1 and the other two components. ARC complex ARG1 and ARB1, ARB2 recognized this siRNA. After they bind to like this. Okay, so ARG1 has the activity to break down the double-stranded siRNA. At that, they will finally make the single-stranded siRNA. Single-stranded RNA is like this. Okay. So ARG1 break down double-stranded siRNA to create single-stranded RNA. So at that, RITS complex is formed. So RITS means that this is RITS, ARG1 with single-stranded RNA and also CHP1, TAS3. So this is RITS, ARG1, TAS3, and CHP1 is RITS, and RITS with a single-stranded RNA forms like this. So this RITS with single-stranded RNA bind to the nascent transcript. Nascent transcript means that 새로 생성되는 transcript. So this transcript is synthesized by RNA polymerase 2. This is a newly synthesized transcript. So RITS with a single-stranded RNA bind to nascent transcript. Actually, at the site that will form the heterochromatin. So they want to form the heterochromatin in this region. So actually, this RITS with single-stranded RNA bind to the nascent transcript at the site that will form heterochromatin. So actually, this RITS is composed of CHP1. So CHP1 is that they recognize methylation. So we'll see some detailed information later. Next slide, okay? So anyway, the important thing is RITS with a single-stranded RNA bind to nascent transcript at the site that will form the heterochromatin. At that, RITS with a single-stranded RNA recruit CLRC. So this is a CLRC. It recruits CLRC. This is also CLRC. CLRC is composed of the CLR4 or RIK1, DOS 1 and 2, and CUL4, clean4. Okay, let's see some detailed information of CLRC. So CLR4 function as this is CLR4. So they are H3K9 methyltransferase. So this CLR4 can make the methylation in H3K9. Histone 3 K is the lysine. Histone 3 is composed of several amino acids. So ninth amino acid is lysine. The lysine can be methylated. So usually it methylated the H3K9 methylation. So usually the CHP1 in the RITS complex So CHP1 is the chromodomain. So chromodomain is also can bind to the methylated histone H3K9, and also it also recruit the SUI6. SUI6 we learned that this is the HP1, or it binds to methylated histone H3K9. So both of them is decodes the information of methylated histone H3K9. So after that, it can initiate heterochromatin formation in this region. Okay. In previous slide, we learned RNAi dependent heterochromatin formation. So let me summarize the RNAi dependent heterochromatin formation. Okay. So this is the model. So RITS with single-stranded RNA can bind to the nascent transcript. Nascent transcript means the newly synthesized transcript. After that, it induced methylation here, and finally it will make the heterochromatin formation here. Okay, so this is, we call it as, I briefly summarized the RNAi dependent heterochromatin formation. This model, we call it as nascent transcriptor model. Okay, so people want to prove this nascent transcriptor model, so people design the experiment like this. Okay, so this is RITS, but they don't have any single-stranded RNA, so they just combined artificial tethering with RITS. So this is one example of the artificial tethering. So this artificial tethering can bind to the mRNA. So by using this artificial tethering, this artificial tethering induces RITS binding to nascent transcript. In this case, this is mRNA. So in these cases, we also observe the H3K9 methylation here, and also we also observe the heterochromatin formation in this region. So this is the experiment to prove the nascent transcriptor model. Okay, we used artificial tethering. So in this RITS, they don't have the single-stranded RNA. This artificial binding is initiated H3K9 methylation and also induced heterochromatin formation. So in this slide, I will compare the function of RNAi in mammalian and the function of the RNAi in pigeon yeast. Let's start from the mammalian. Okay, so in the mammalian, after double-stranded RNA was synthesized, after that, several processes happens, so they will make the risk complex. The risk complex, they have the single-stranded RNA. Okay, so but the risk complex is working in the cytosol, not nucleus. So this RIC risk complex bind to specifically to mRNA sequence. So this binding induces sequence-specific RNA degradation. So it means that it will induce post-transcriptional gene silencing. How about in pigeon yeast? In pigeon yeast, we already know that RITS complex is located within nucleus. So this RITS with single-stranded RNA bind to the nascent transcript, and after that it will induce the H3K9 methylation. After that, finally it will induce post-transcriptional gene silencing, PTGS. Okay, and also especially in the Fusion Yeast, this PTGS, we call it as the cis PTGS. We will see some details in the next slide. So cis PTGS means that cis acting post-transcriptional gene silencing. Okay, this is the difference between cis and trans. So cis is working within one chromosome. Okay, trans is working occurs in different chromosome. For example, this is a cis PTGS. For example, link RNA is the transcribed nearby gene A. So this transcribed link A bind to the factor X. After that, it will affect gene expression of gene A, nearby gene A. So sometimes it can activate expression of gene A, or sometimes inactivate the expression of gene A. So it happens the nearby chromosome. So it means that this is we call it as the cis-acting. Trans-acting means that link RNA is transcribed from different chromosome, so it moved to another chromosome. After that, this one bind to another factor Y and also affect the gene A, which is located in the different chromosome. So this is trans-acting. However, the RNAi dependent heterochromatin formation. So this RNAi dependent heterochromatin formation usually always occurs nearby heterochromatin. Because in the nearby heterochromatin, if there is a nearby heterochromatin, after that it will induce the RNAi dependent heterochromatin formation. Okay, so it means that in this region is that, okay, for example, this region was in the heterochromatin region. So after that, it will synthesize siRNA. After that, the RITS complex is made. So after that, this complex is bind to the nearby chromosome. After that, it will recruit another CLRC complex. After that, it will induce H3K9 methylation. So this means that RNAi dependent heterochromatin formation is cis PTGS. So there is another concept, TGS. TGS, we call it as transcriptional gene silencing. TGS do not use RNAi. So for gene silencing, TGS do not use RNAi, but uses other compounds. Okay, let's see one example. So this is one example of the SHREC. We call it the Shrek complex, Shrek protein. So this protein inhibit RNA polymerase binding to promote region. So it inhibits RNA polymerase binding to promote region. So it will induce decreased RNA synthesis. So it means that decreased RNA synthesis means that the gene silencing. So in these cases, we call it as the transcriptional gene silencing. It do not use RNAi. It just inhibits RNA polymerase binding to promoter. So simultaneously gene silencing happens. So this is a TGS. Okay. In this slide, I will explain the fifth subject, spreading and establishment of heterochromatin by RNAi. So in this slide, I will explain some detailed mechanism of the maintenance of heterochromatin during the cell cycle. So cell cycle is composed of G1, S, G2. This is the interphase. After interphase, there is a mitosis. Mitosis happens like this. Okay. So let's start. So in the Fusion Yeast, G2 phase is comprise 70% of the cell cycle. Okay, so let's start from the G2 phase. So during S phase, DNA is synthesized and one chromosome becomes the two chromosome. Okay, so during G2 phase, each pericentric heterochromatin is enriched in H3K9 methylation. So each pericentric heterochromatin is enriched in H3K9 methylation. So this is a histone methylation marks. So this is H3K9 methylation. So usually this methylated site provides a binding site for SUI6 protein. So this is SUI6 protein bind to the methylated H3K9. Okay. So after G2 phase, so there is a mitosis happens. So during mitosis, usually so this is the histone H3 histone. So this is in this case is the H3K9 methylation is happen. So in this H3 histone is composed of lots of the amino acid. In 9th location there is the K means that lysine. In 10th position there is S means that serine. So in the 9th location was the methylated, but during M phase, 10th location, serine is phosphorylated. The serine is phosphorylated. So during the M phase, serine is phosphorylated. So it means that phosphorylation of the H3S10, especially in 10th location, serine is phosphorylated. So this is lead to separation of SUI6 from methylated H3K9. So during the S phase, G1 phase, after finished S phase, mitosis, they will go to the G1 phase. So G1 phase is to show that the status is that most of the H3 histone is phosphorylated, especially at 10th serine locus. But some of them is not phosphorylated. So in this case, SUI6 is bind to this region. Okay. So during the S phase, after they finish G1 phase, they go to the S phase. So during the S phase, DNA replication is happen in the pericentric heterochromatin region, and also transcription is happens in pericentric heterochromatin region. So after that, after transcriptions, non-coding RNA is synthesized. At that, it will make the several processes there. At that, it will make the siRNAs. So we already covered all of the procedure in previous slide 1, 2, 3, 4. At that, it will recruit CLRC complex. In CLRC complex, they have the CLR4. This CLR4 main function is that H3K9 methyl transferase. So it means that this CLR4 is the induced H3K9 methylation in newly synthesized nucleosome. So this methylation induced heterochromatin formation in both pericentric heterochromatin. So after the finished S phase, so they will go to the G2 phase. So in G2 phase, so all of both pericentric heterochromatin is enriched in H3K9 methylation. So in this slide and this slide, we cover the maintenance of heterochromatin during cell cycle, starting from the G2 M phase at that the G1 S phase. So during the after transcription in the pericentric heterochromatin, it happens after that it will make the siRNA. Some specific mechanism we already learned in this the slide. Finally, the CLR4 function as the H3K9 methyltransferase. So it induced H3K9 methylation in newly synthesized nucleosome. So finally, it induced heterochromatin formation in both pericentric heterochromatin region. Okay. So in this slide, I will remind Rna dependent heterochromatin formation mechanism. So usually the bidirectional transcription is happen in the OTR region of the centromere. For example, the DH and DG sequence is composed of the OTR region of centromere. Okay, so after that, it will generate non-coding RNAs. One is the sense, the other is an antisense, the non-coding RNAs. After that, exosomes removes most of the non-coding RNAs. But three men bind to the remained non-coding RNA. So actually this three-man protein is protect from the exosome. So it means that it can protect the remained non-coding RNA, and after that it processes RNA, so it will generate pre-RNAs. After that, ARG1 protein bind to sense or the antisense pre-RNA. So this is, we call it as ARG1 pre-RNA complex. So in this slide, I will explain the RNA quality control system and heterochromatin formation. So usually after non-coding RNA is synthesized, the exosome is removed the non-coding RNAs. They digest it and it removes non-coding RNAs. So when we can remove the exosome, if we can remove the exosome, it means that three-man can synthesize the more pre-RNAs. It means that if there is more pre-RNAs, it means that it will promote heterochromatin formation. Or after pre-RNA is synthesized, usually the cell has the mRNA export mechanism. So synthesized RNA transcript can be actively transported into the cytosol. Usually the synthesized transcript or synthesized mRNA can be exported outside of the nucleus. So they will go to the cytoplasm. So if we remove the mRNA export mechanism, so it means that pre-RNAs stays longer in the nucleus. So it means that the effect is that the cell has more pre-RNAs. It means that this mechanism also promotes heterochromatin formation. Okay? So in this slide, I will explain the final subject, facultative heterochromatin in fusion yeast. Heterochromatin is composed of two subtypes. One is constitutive heterochromatin and the facultative heterochromatin. So this is constitutive heterochromatin. This is this one has the repeated sequence, and we can always observe the constitutive heterochromatin in the centromere region and telomere region. And also, usually in this region, we already see that RNAi mediated heterochromatin formation is happen, and also in centromere and telomere region. So what is the facultative heterochromatin? So facultative heterochromatin is they can convert to euchromatins under certain conditions. Okay, but this facultative heterochromatin do not have a repetitive sequence. So usually the gene locus involved in the meiosis and also gene locus involved in mating, especially for example, the mating type locus. This gene is within the facultative heterochromatin, so their location, maybe they are located in the within this region, some kind of this region here, somewhere in this region. Okay. So in this slide, I will explain one example of the facultative heterochromatin. The name is the Heterochromatin Islands. So gene locus involved in meiosis exists within Heterochromatin Islands. So this region we call it as Heterochromatin Islands. So actually the DSR core sequence. So DSR, this is not repetitive sequence. They have the DSR core sequence. For example, UCAAAC or UUAAAC. So usually in the Heterochromatin Islands, so the DSR core sequence is existed. For example, Meiotic gene also has this DSR core sequence. So in this island, this heterochromatin island, so require the MTREC protein, and also it require exosome to make the heterochromatins. But in this slide, I will not explain some detailed mechanism of heterochromatin formation in these heterochromatin islands. So in this MTREC protein and also exosome to make heterochromatin formation. Okay, and also another example is hood. Hood is that heterochromatin domain in facultative heterochromatin. So hood, we call it as hood. So hood is that we also have some sequence, domain sequence. So in this domain, gene locus involved in mating. So for example, mating type locus existed within hood region. Okay. So hood region, hood requires RNAi and MTREC and exosome to form heterochromatin. But in this slide, I will not cover in detail about the heterochromatin formation mechanism in hood. Okay. So just briefly, gene locus involved in mating, for example, mating type locus exists within hood, and also hood requires RNAi and MTREC and exosome to form a heterochromatin. Okay. Today we learned about Chapter 3. Chapter 3 title was Epigenetics in Fusion Yeast. First, we learned the Fusion Yeast life cycle. We also learned the Centromere Structure, and also we learned the heterochromatin formation mechanism in centromere, and especially we also learned about RNAi dependent heterochromatin formation mechanism. We call it as a cis-PTGS. And we also learned about the heterochromatin spreading and maintaining mechanism. And finally, we learned about facultative heterochromatin. So today we will finish the class.